Welcome back, everybody, for another deep dive. Today, we are going to be looking at cellular senescence, which is basically how our cells age. We're going to be taking a look at this paper from the journal Science Advances called Mitochondrial Fatty Acid Oxidation Drives Senescence.
Yeah, that's a pretty dense article. We're going to try to break it down for you.
Yeah, from what I've been reading, it goes way beyond wrinkles and gray hair. It actually gets into like DNA damage and then these little things called mitochondria in our cells, and even how our bodies break down fat.
You're right. We are really diving down deep to the molecular level here. So to start, let's just get a clear picture of what cellular senescence is. Basically, it's when cells stop dividing.
So they're not dead, but they're not exactly thriving either.
Exactly. Think of it as like the cell hitting the pause button. And this is often triggered by stress and damage to the cell, particularly to the DNA. And as you can imagine, over time, that damage builds up and cells go into senescence.
So damaged cells going dormant? That almost sounds like a good thing, like the body's way of preventing further problems?
You're onto something. Senescence actually acts as a tumor suppressor. So by stopping these damaged cells from multiplying, it can help prevent them from becoming cancerous.
Oh, that's amazing. So it's linked to aging, but it also protects us from cancer. Talk about a double-edged sword. I think what I find really interesting, though, is the focus on mitochondria in this study.
Yeah.
I remember learning about these as the powerhouses of the cell, but I have a feeling there's a lot more to the story.
Absolutely. While it's true that they're crucial for producing energy, they also play a significant role in cell signaling and really determining the cell's fate, whether it lives or dies or enters this senescence. And what this study does is it really sheds light on a specific mechanism linking mitochondria to senescence.
Oh, so they kind of zoomed in on one piece of the puzzle. What did they find?
So they identified this protein called BNIP3. It's located on the outer membrane of the mitochondria. And through this massive genome-wide screen, they found that BNIP3 is crucial for this DNA damage-induced senescence.
Wait, a genome-wide screen. That sounds incredibly thorough. So they basically scanned the entire genetic blueprint to find this one protein.
That's right. It really highlights the scale and the depth of this research. They weren't looking for a quick answer. They were determined to find the key players. And BNIP3 turned out to be this critical link between the DNA damage and senescence.
Okay, so BNIP3 is important?
Yeah.
But how does it actually work? What is its role in this whole process?
Well, so when DNA damage occurs, it triggers a cascade of events within the cell. One of the first responders is this kinase called ATM, and this usually operates in the nucleus, the cell's control center. But here's the surprising part. ATM can also travel to the mitochondria and activate BNIP3.
So there's communication happening between the damaged DNA and the nucleus, and this protein on the mitochondria. That's wild. So what happens once BNIP3 is activated?
So once it's activated, BNIP3 goes to work changing the structure of the mitochondria. Specifically, it increases the number of cristae. Those folds inside the mitochondria where a lot of that energy production is happening.
Right, the cristae. More surface area for energy production makes sense. But how does that actually lead to senescence? Changing the structure of the mitochondria.
This is where things get really interesting. By increasing cristae, BNIP3 enhances this process called fatty acid oxidation, or FAO. You might be familiar with FAO if you've ever heard of the keto diet. That really emphasizes fat as a primary fuel source.
Oh yeah, the keto diet.
Yeah.
So we're talking about how our bodies break down fat for energy. I'm still trying to connect the dots here, though. How does FAO fit into this whole picture of senescence?
Well, it turns out that increased FAO can lead to higher levels of this molecule called acetyl-CoA. And this molecule plays a really critical role in a lot of different cellular processes. One of its key functions is modifying histones.
Histones. Those are the proteins that the DNA wraps around, right? Like spools for thread.
Exactly. That's a great analogy. Acetyl-CoA can attach to these histones, a process called histone acetylation. And this can actually change how that DNA is packaged and how accessible genes are.
So like changing the way the DNA is organized.
Yeah.
And then that affects which genes are turned on or off. So to see how this all comes together.
Exactly. And in this case, they found that this increased FAO through BNIP3's action on the mitochondria leads to more histone acetylation around a specific gene, the one that codes for P16.
Wait, P16, didn't you mention that earlier?
Yes. P16 is a key protein involved in senescence. Basically, it acts like a brake on cell division, and its levels naturally increase in our tissues as we age.
So what we're seeing here is this whole chain reaction. DNA damage triggers this cascade, changes the mitochondria, boosts this FAO, and then ultimately ramps up the production of a protein that drives senescence, like a domino effect at the molecular level.
That's a great way to put it. And what's even more fascinating is that they found that they found that activating FAO artificially, even without DNA damage, can actually induce senescence.
Hold on. That's a huge claim. You're saying they can make cells age prematurely just by manipulating this one process. How is that even possible?
Well, so they did it using two different methods. First, they treated the cells with octanoate, which is a type of fatty acid that directly enters the mitochondria and boosts FAO. And sure enough, it triggered all of these changes in the cells that look very similar to what you see in natural senescence.
So they basically flooded the cells with this fatty acid, and they started seeing signs of aging. What was the other method?
They used a drug called fenofibrate, which is already known to activate FAO. And again, they saw these senescence-like changes in the cells.
Wow, so this suggests that FAO isn't just a consequence of DNA damage, but it could be a driver of senescence all on its own. It's a pretty radical idea. Are you saying that, like, our diet could actually speed up or slow down cellular aging?
It's certainly a possibility. You know, this research really highlights this very specific pathway. DNA damage, ATM activation, BNIP3 in the mitochondria, increased FAO, and then ultimately P16 expression in senescence. And if we can really understand all of the steps in this pathway, we might be able to find ways to influence it.
That makes sense.
Yeah.
But I'm thinking, isn't FAO also crucial for energy? Our bodies need to break down fat for fuel. How do we balance that need with the potential downsides of, like, accelerating senescence?
Right. It's all about striking that balance. You know, completely shutting down FAO would be really detrimental. But maybe there's a sweet spot, a way to fine-tune FAO to really optimize both cell health and longevity. It's a question that researchers are actively investigating.
Okay, so let's rewind for a second. You mentioned that this increased FAO leads to higher levels of acetyl-CoA, which then affects histone acetylation and ultimately turns up the volume on that senescence gene P16. But I'm still a little fuzzy on how that actually works. It seems like kind of a big leap from breaking down fat to controlling gene expression.
It is a complex process, but you can think of it like this. Acetyl-CoA is like a cellular messenger. It carries these little chemical tags called acetyl groups that can attach to different proteins, including histones.
So we've got these tags attaching to the histones. Those protein spools that the DNA wraps around, what difference does that make?
Well, those tags can actually change how tightly that DNA is wound around the histones. You can imagine like a tangled ball of yarn. If you start to loosen some parts of the yarn, it becomes easier to access certain sections.
Okay, I'm visualizing it. So by adding these tags, you're basically loosening up certain parts of the DNA, making them more accessible.
Exactly. And that accessibility is key for gene expression. You know, it determines which genes are read and used to create proteins. And in this case, by increasing acetyl-CoA through FAO, you're essentially making that P16 gene more accessible, which leads to increased production of that P16 protein. That drives senescence.
That's incredibly elegant, but I have one more question. You mentioned that acetyl-CoA is made in the mitochondria, but histones are in the nucleus. So how does the acetyl-CoA actually get from the mitochondria to the nucleus to do its tagging?
That's a great question. It's almost like a cellular postal service. The acetyl-CoA actually needs a special package to get out of the mitochondria and reach its destination in the nucleus. So it first converts into a molecule called citrate, which can actually cross the mitochondrial membrane.
Ah, so it hitches a ride out of the mitochondria, disguised as citrate. Very clever. And then what happens?
Once the citrate reaches the cytoplasm, that's the area outside the nucleus, it gets converted back into acetyl-CoA, and from there it can enter the nucleus and attach those acetyl tags to the histones.
Wow, so it's like a whole intricate transport system just to deliver these tiny tags. But back to the bigger picture. If they found that activating FAO artificially can induce senescence, does that mean that we could potentially slow down aging by doing the opposite, like reducing FAO?
Now, that's a question that researchers are actively exploring. But remember, FAO is a fundamental process for energy production, especially when our bodies need to burn fat for fuel. You know, completely shutting it down would have serious consequences. Think about it like when your car runs out of gas, everything grinds to a halt.
Right, it's all about finding that balance again. But the fact that we're even talking about the possibility of influencing aging through something like diet or medication is just mind-boggling.
It really is a fascinating frontier, and I think this research has opened up so many avenues for further exploration.
Speaking of exploration, I'm curious about the implications of all of this. We've been so deep in the molecular weeds here. But what does this research actually mean for treating age-related diseases?
Yeah, this research is definitely making me think differently about what we eat and how we age. But before we get too carried away with like future possibilities, what are the next steps in this research? What are scientists grappling with now?
Well, there's still so much that we don't know. For starters, we need a much more detailed understanding of how BNIP3 actually alters the mitochondrial structure. We know that it increases crystio, but how does it do that? What other proteins are involved? What are the specific steps in this whole remodeling process?
And how does that structural change actually boost FAO? Is it just more surface area, or are there other factors at play? Right. It's like we have a puzzle and we found a couple of key pieces. But now we need to figure out how they all fit together. And then, of course, there's the big question of how exactly increased FAO actually drives senescence. We know it leads to higher acetyl-CoA levels. But are there other mechanisms involved?
You mentioned earlier that increased ROS production could be a factor. Could you explain that a bit more?
Sure. So reactive oxygen species, or ROS, are basically byproducts of cellular metabolism. Think of them like exhaust fumes from a car engine. A certain amount of ROS is normal and even necessary for things like cell signaling, but too much ROS can damage DNA proteins and other cellular components.
It's like the cell zone pollution problem.
Exactly, a good analogy. And some research suggests that increased FAO can actually lead to higher ROS production in the mitochondria, so it's possible that this increase in ROS is contributing to that cellular damage that ultimately drives senescence.
Wow, so FAO provides energy, but it could also be contributing to cellular aging through ROS. Talk about a delicate balance. But let's circle back to those potential applications we touched on earlier. You mentioned treating age-related diseases. Are there any specific areas where manipulating FAO seems particularly promising?
Well, one area that comes to mind immediately is cancer. Cancer cells have a very different metabolism compared to healthy cells, and some types of cancer really rely heavily on FAO for their growth and survival.
Oh, so if you could target and reduce FAO in those cancer cells, you could essentially starve them.
Exactly, that's the idea. And researchers are actually actively investigating that strategy. In fact, there are already some drugs that inhibit FAO that are being studied in clinical trials for cancer treatment.
That's incredible. What about other age-related conditions like heart disease or Alzheimer's disease? Could manipulating FAO play a role there too?
Well, there's growing evidence that mitochondrial dysfunction is involved in both of those conditions. And since FAO is a key mitochondrial process, it's definitely a potential target for intervention.
So by understanding how FAO contributes to cellular aging, we might be able to develop therapies that protect the mitochondria and prevent or delay the onset of these diseases. It's like we're starting to see a whole new approach to medicine, targeting these really fundamental processes within our cells.
Exactly. It's all about connecting the dots and understanding this intricate web of molecular processes that govern aging. And this study has provided us with a really crucial thread to follow.
This has been a mind-blowing deep dive. We've gone from the basics of cellular senescence to the intricate workings of mitochondria, and we've even touched on the potential for manipulating aging.
Yeah.
And treating age-related diseases. I feel like I've just taken a crash course in cellular biology.
Well, it's been a pleasure exploring this fascinating research with you, and I really think this is just the beginning. There's so many more exciting discoveries to come.
I couldn't agree more. And for our listeners out there, I want to leave you with this thought-provoking question. This study focused on how DNA damage triggers FAO and senescence. But what about other factors that we know contribute to aging? Things like chronic inflammation or oxidative stress. Could they also be influencing this pathway?
That is a great question, and one that researchers are undoubtedly investigating as we speak. The science of aging is constantly evolving, and there are countless mysteries still to unravel.
So to all of you listening out there, keep those curious minds engaged, and stay tuned for more deep dives into the world of science.
Until next time, keep exploring and keep learning.